Welcome to the tutorial video series on the GA4GH Variation Representation Specification, or VRS, pronounced VERSE. This is the first in a series of short tutorial videos to learn about VERSE and will serve as a lightweight introduction to the specification. Hi, my name is Larry Babb, and I'm a software engineer at the Broad Institute in Cambridge, Mass. I'm also co-lead of the Variation Representation subgroup and a ClinGen driver project champion for GA4GH.
So my name is Alex Wagner. I co-lead the variation representation team with Larry. I'm an assistant professor at Nationwide Children's Hospital in Columbus, Ohio. And I'm also the driver project champion for the Variant Interpretation for Cancer Consortium. So with introductions out of the way, let's go ahead and jump into the specification. Great,
thanks, Alex. All right, the need for VERSE, a standard for variation exchange, is not new. Today, there are dozens of methods implementers use to exchange variation for both structures, nomenclatures, minted public and/or private identifiers. As we will see, these approaches eventually run into challenges when systems begin taking on the challenge of mapping between them in order to associate related data. And that's when data is missed and incorrectly related. It creates misinformation, which is certainly detrimental to the aims of applying genetic research and analysis in precision medicine initiatives. So where does VERSE fit in the big picture? One of the major motivations behind the need for VERSE is to reduce the inconsistency and time required to evaluate and report variant findings in genetic testing, known as the variation evaluation bottleneck. The individual in the middle of this figure could be a molecular pathologist, a geneticist, a curator, a clinician, or even a variant scientist. The real challenge is connecting the biomarkers to publications and heterogeneous data sets and knowledge bases. This requires human expertise and is very time consuming.
So what do we mean by heterogeneous data sets? Well, consider how, for example, you might go about a genotyping study. So yeah, when you're trying to do genotyping, you're looking at biomarkers that basically indicate small differences in a potential genome compared to some reference. And that genotyping exercise is typically done through some type of assay, usually something like a high throughput short read sequencing assay shown here. And from the output of that assay, you then might go and algorithmically generate variant calls from those sequencing reads. And as a result of this process, you'll end up with like a variant call format file that will contain all of those small variants, those SNVs and indels, with respect to your reference sequence, and that represents your genotype. But the fact is that genotyping is one of a set of highly relevant, clinically and research relevant types of biomarkers that you may choose to assay. This is not an exhaustive set of those biomarkers. And it can be, and any of these can be assayed by a number of different related technologies. And each one of those technologies may have multiple ways by which the outputs from that technology are interpreted and turned into observed variation, which is represented in a variety of different formats. And so you end up with all these heterogeneous ways of describing variation that relate to all these different types of biomarkers assayed under any of these technologies, and then called using any of a number of different methods for what is effectively an exponential set of pathways by which you create observed variation.
Wow, Alex, that's something. And the challenge is only compounded by the fact that there's a myriad of resources with mostly different forms of identifiers that are expected to be used to connect the observed variation to the knowledge and evidence collected in these resources. While some of these resources, like ClinVar variations and ClinGen allele registry canonical alleles, are structured similarly, even though they have different identifiers and capabilities for public registration, others, like somatic cancer variant resources of Civic and Cosmic, cross-reference these variation identifiers from related but distinct variation concepts.
This is kind of like having a Civic variant that references a ClinGen allele registry CAID.
That's right.
Yeah, and so the idea then is, in order for us to be able to do those types of mappings better, we want to be able to get from these various forms and flavors of variation into something that has well-structured, well-defined, computable semantics. And that's what verse is ultimately designed to do. It allows us to express the semantics. And importantly, it allows us to then take those computable representations of variation and use that to interpret computable representations that exist in genomic knowledge bases, and ultimately be able to computationally assemble reports into a database that ultimately get reviewed as opposed to assembled by your clinical expert, be it a variant scientist or clinician or whatever. And this can greatly speed up the process of variant assessment and interpretation.
Great. So now let's move on and talk about how verse really not only fits in, but how it compares with some of the existing standards and tools in the ecosystem. If you're watching this tutorial, you are likely familiar with VCF and HGVS variant formats. These formats serve an important role for particular use cases, such as high throughput variant calling as captured by VCF, and human readable variant descriptions as represented by HGVS nomenclature. These live alongside registered variant identifiers like those found in dbSNP, ClinVar, Civic, ClinGen Allele Registry, and others. Due to the differences in scope and purposes of these formats, it can sometimes be challenging to map across them, oftentimes impossible. These examples below each format represent different variant contexts associated with the BRAF V600 variant, for example. Verse aims to improve the computational exchange of simple to highly complex forms of variation. The GA4GH Genomic Knowledge Standards Workstream assembled driver projects and individuals to establish verse and begin extending and maturing it with tooling and system implementations so as to broaden its adoption and realize its aims. Verse picks up where the variant calling leaves off. It does so based on minimalistic data structures, which are designed to significantly increase the interoperability and linkability of data sets that contain variation from heterogeneous sources. You can find the specification online at verse.ga4gh.org. With verse, we are aiming to move from the ecosystem on the left to the one on the right. The ecosystem in the before panel represents how Exchange typically works today, requiring each system to parse, map, align, and otherwise transform variation formats from other systems into a localized, coherent form. In the after panel, verse provides genomic data systems with an interoperable computable framework for exchanging their own variation concepts, reducing the risk of misinterpreting the variation concepts shared and consumed. VCF, HGVS, and other potential representations available today are not designed for the breadth and complexity of variation concepts necessary for reproducible and interoperable data exchange across this diverse landscape of genomic data resources.
So verse as a standard is really a series of components that are interoperable and built upon one another. Starting with the core of a terminology and information model, verse also defines standard conventions and protocols for the exchange of variation objects. Verse has a reference implementation in Python, which includes tools for transforming variants in genomic data systems, shown here in green boxes, from other common formats such as HGVS or SPEEDY or VCF into verse. However, the core information model verse might also be used with alternate protocols or conventions. For example, the GA4GH phenopacket standard leverages a protocol buffer representation of the verse information model, while the GA4GH beacon standard uses the native JSON schema. So speaking of the verse core terminology and information model, there are really four major groupings of data classes that we are concerned with. And each of these has abstract types associated with it, allowing for object inheritance and or property inheritance in data classes. The first of these is variation. So variation is, of course, the core of what makes the variation representation specification. It includes molecular variation concepts such as allele and haplotype, as well as systemic concepts such as copy number variation. To be clear, the classes indicated here are not exhaustive. There are other variation classes that exist in verse, as there are other sequence expression classes, other supporting data types. But the point here is that we want to provide an example of the major groupings of the types of entities we have, and the notion that many of the variation expressions that you wish to make can be composed from these underlying fundamental types. Now, moving over to location, location is a fundamental property that we think of when we're talking about sequence variation. Sequence locations are kind of the standard or canonical way of thinking about how variation sits on a sequence. Chromosome locations, in contrast, are more like a cytogenomic representation of a coordinate system. So think like karyotypes and cytobands. But both coordinate systems can be used in the representation of variation within VRS. Sequence expressions are a way of representing sequences, either in a literal format with literal sequence expressions, where you have A's, C's, T's, and G's, or amino acid residues that are composed as a string that describe the sequence. Or it can be something as abstract as derived sequence expression, where you provide a sequence location or other location object, and you can use that to say, I'm talking about the sequence that may have been derived from that location. And then finally, there's a variety of different supporting data classes, things like genes and numbers and ranges that allow us to create these higher order constructs.
Thanks, Alex. Now we're going to spend the next 3 slides just touching on a few key design decisions without going into great depth, so that we can bring it all together and show how we deal with something like molecular variation. One of the foundational design decisions in VRS was to use an inter-residue coordinate system in lieu of the residue coordinate system used by the VCF and HGVS formats. We selected this approach to help disentangle variant operations from sequence coordinates. To illustrate this, let's compare how insertions at the top or deletions and substitutions at the bottom are handled in each of these coordinate systems. Residue-based coordinate systems, gray highlighted coordinates in these diagrams, exclude residues in the coordinate range when describing sequence insertions, but include those residues when describing substitutions or deletions. In contrast, inter-residue coordinate systems in the blue highlighted boxes are always inclusive and can be placed precisely and unambiguously between residues in sequence insertion contexts, thus decoupling the variant type from the choice of coordinates.
The other big convention that we have selected in VRS is how we choose to normalize variation. So this particular concept is really due to the way that we profile sequence changes compared to a reference. So when you do a sequencing experiment and you make that comparison, you're measuring some end state. And that end state basically says, at this given position where I maybe had 3 C's before, I might have 5 C's. Whereas in this example, there's a CAG repeating sequence. Maybe you have an additional CAG, or maybe you lose a CAG. And so when that happens, you need to be able to represent, okay, well, where did that occur? And because of the ambiguity, and because we're only observing the end state, we don't actually see where this event happened, we need to basically select by convention how to represent this variation. And in standards like VCF and HGVS, they pick an arbitrary convention, either left shuffling, which is what VCF does, where they put the CAG insertion all the way to the left of the repeating sequence, or they can shift all the way to the right, or the 3' rule, as expressed in HGVS, where we see the AGC is placed at the very rightmost edge of the repeating region. But what VRS does is it actually takes the entire region of ambiguity and replaces it with a slightly larger region, including the additional nucleotides. So we're being exactly as precise as our assay technology allows us to be. And what that also means is that we're now overlapping those genomic coordinates, and we don't get this challenge of having to represent the sequence in different ways, depending on if we pick left or right. And we don't run the risk of entirely missing overlaps of those sequences because of that choice.
Another key principle behind the design of VRS is value object representation. So what do I mean by that? This principle requires verse data structures to be minimal representations containing only the most salient definitional elements necessary. Achieving the value object constraint enables genomic data systems to use these components as self-identifying.
And in a practical sense, what that means is that this singular representation at the data level of that variation can be leveraged in order for us to do some cool stuff. And one of those things that we can do is developing computed identifiers. So by representing verse variation as value objects and going through a series of computational steps, we're able to convert those alleles and other forms of verse variation into identifiers via computational digests. And what that means is the various expressions shown here in panel D that are used to describe variation, and I use different reference sequence, et cetera, they all digest down to the same computed identifier. And so if you have the same change on the same underlying sequence, regardless of what the label of that sequence is, you're going to end up with the same variant identifier for that change. And what's powerful about that is we're now able to create globally unique computed identifiers from anywhere in the world without prior negotiation.
These computed identifiers provide a tool to help realize the promise of interoperable system exchange, as illustrated by the keys in the after panel shown here. As we have demonstrated, verse is a value object-based precise representation of variation. It does not provide for additional annotation, metadata, etc., to be exchanged. To be pragmatic, verse needs to be extended to enable implementers the freedom to share additional attributes and decoration around variation. So we have created a complementary extension to verse called VRSATILE, which is the verse added tools for interoperable loquacious exchange. In essence, VRSATILE provides the exchange wrapper for verse components, allowing implementers to share descriptive variation metadata. Please visit the VRSATILE spec site to learn more about it.
So that concludes our introductory overview of the GA4GH variation representation specification. If you would like to provide any feedback on the contents of this tutorial, what you've learned, what we might have done better, please feel free to do so at ga4gh.org/verse-feedback. Additional tutorials are planned for Verse Python, for VRSATILE, and for some of the nitty-gritty technical details around verse. Also, feel free to go out and read the verse standard and the VRSATILE extensions at the URL shown here. Finally, feel free to try out verse. We have some cool beta production cloud-based notebooks you can go check out at go.osu.edu/verse-cloud-nb. Or you can go just look at those notebooks yourself, download them onto your local machine and give it a try, looking at github.com/ga4gh/verse-python. Thanks everyone for your time and interest in verse, and we look forward to seeing you in our next video.